My name is Ben. I'm from Liam Paninsky's group, and I'm going to talk a little bit today about reconstruction and denoising of dendritic voltage from voltage imaging experiments. Huge thanks to the organizers. This has been really fun. And so to motivate this talk, I'm going to start off by showing this figure from Nelson Spruceton. It's a classic paper where they managed to patch both the soma and the dendrite of a CA1 pyramidal cell. And they found dendritic spikes that were ablated by TTX, meaning that these dendritic spikes required voltage-gated sodium channels. And so dendrites are active participants in computation. They're not just passive propagators of voltage. And there are still a lot of unknown questions about what contributions dendritic computation makes. In particular, we'd like to know how dendritic phenomena arise, what conditions must be true for the cell to generate these, and we also would like to know whether the whole tree gets the same signal. You can imagine that that second question would be very important if you're interested in plasticity. And it's difficult to study that second question if you're just doing patch experiments, but thankfully a lot of very hardworking experimentalists have developed genetically encoded voltage indicators. And these now allow for voltage imaging of dendritic trees. I'm showing a figure from our collaborators in Adam Cohen's lab. But there are some computational advances that are needed to take full advantage of these experiments. So dendritic voltage events happen very quickly, on the order of 2 to 3 milliseconds. And so that means that we need to image very fast, around 1 kilohertz or even faster, to capture these. And in microscopy, there's a fundamental trade-off between the imaging speed and the field of view. If you imagine that I have a dendritic tree here, and I want to scan a laser across every point in 3D space, that's probably going to take longer than a millisecond, although there are some clever hacks. And so volumetric imaging, 3D imaging of this dendritic tree, is currently very difficult at 1 kilohertz. And we've been working with collaborators in Adam Cohen's group, and they have designed a very clever series of experiments that we think gets around, or somewhat gets around, this speed FOV trade-off. The way their experiments work is they do 1 photon wide field imaging of the dendritic tree. And then later on, they go in with a different microscope, and they reconstruct the anatomy of the tree using either 2-photon or confocal. And if we imagine that there's some 3D signal at all of the orange points, there's some voltage signal in 3 dimensions, there's some transform between that 3-dimensional signal and the 2-dimensional voltage frame that we get to observe in the movie. And that transform will be given by the point spread function, or PSF, of the microscope. The point of my talk is that I'll hopefully convince you that we can go backwards from this 2-dimensional voltage frame and recover a 3-dimensional voltage signal. And now this is all assuming that we know the anatomy of the cell in advance. Without that, I don't think this is possible. And so I'll start off and I'll show a little bit about what these data look like, because the data themselves are really cool. On the left, there is hopefully, yeah, there it is, a scan through Z of the 2P anatomy stack. And on the right is a voltage movie. And what you'll see is, like 2/3 of the way through, you should see a spike starting at the soma and propagating back into the dendritic tree. I think looking at these movies is really fun. And so using these 2 types of data, our goal is to reconstruct this 3D voltage signal. We start off and we pre-process the movie with PMD, which is a lightweight SVD-based denoiser. And we designed a pipeline to go from that pre-processed movie back to the 3D voltage signal. And it's very unfortunate that I have to introduce another acronym, because there have been so many acronyms today. This one we called it DENDRO, for dendritic denoising and recovery. Acronyms are not my strong suit. There's 2 steps to this. The first is that we used the knowledge of the optical system to solve a linear inverse problem separately for each frame. But that's very dumb, because that doesn't exploit any temporal structure at all. And so the second step is that we denoise the noisy estimates from step 1 by training a self-supervised neural network. And if everything goes according to plan, I guess I'm allowed to walk around. if everything goes according to plan, we end up with this 3D voltage signal at the end where we can look at different frames of the movie and see the spikes propagating back into the dendritic tree. Cool. So now I'm going to zoom in on the first step of our pipeline, which is solving a linear inverse problem separately for each frame. We're going to imagine that there are some node activity weights that are going to tell us the relative voltage signal at different points along the tree. And I know this is very abstract for now, but our goal is going to be to relate these time-varying signals to the fluorescence that we observe in the movie. In order to do that, we take the anatomy stack that we've gotten from the experiment, and we lay down a graph. And this graph is shown by the dark gray circles and lines. So each dot there is a node in the graph, and the edges are the lines. At each node in that graph, we're going to lay down a smooth basis function. These are the orange things. I'm showing two of them. And the idea here is that at a given frame, we'll form a linear combination of these smooth 3D basis functions, and that will give us a smooth 3D signal. Again, I'm showing this just for a certain frame here, but we'll have a separate vector of W's for each frame. So W's are node activity weights. If you were to take all of the W's into a matrix, you'd have nodes by time. But for now, we're just going to solve one frame at a time. And we're going to estimate a vector of node weights, one weight for each node in the graph. Now, once you've formed this smooth 3D signal, you can pass it through the microscope matrix M, and that's linear because it's just doing a convolution. What the microscope matrix does is it blurs the parts of the cell that are away from the focal plane, and then it adds everything together across Z. I meant to say that earlier. Okay, that's the idea. If we write all this down, we get a simple linear equation. B times W, B is the basis matrix. So the basis functions are along the columns of B. B times W is this smooth 3D signal. And we pass that through the microscope transform M. And we hope that that will be approximately equal to our observed movie frame. I know I'm going very fast, sorry. And so now what we've done is we've estimated these node weights W for each frame of the movie, but we haven't used any temporal information. We want to do some sort of smoothing over time, but the smoothing problem is very difficult because voltage follows these complicated nonlinear equations that are similar to Hodgkin-Huxley models. And previous approaches, Scott actually wrote a very nice paper about this in 2019, used sequential Monte Carlo, or SMC, which is going to be difficult to scale up to our data sets. Each data set that I'm going to show has 10,000 frames. I hope Scott doesn't get too mad, because I know he's a big SMC fan. Anyway, to illustrate what makes this smoothing problem hard, here's a multi-compartment model simulation. The units here are fluorescence rather than voltage. And if we look at sub-threshold regions, the voltage changes smoothly both in time and space. But during a spike, the voltage changes much faster. And so linear methods like a Kalman smoother won't be able to handle this very well. To handle these two regions of smoothness, we used a type of neural network called a blind spot network. These networks have previously been applied to voltage and calcium imaging movies. The way it works when you apply it to a movie or an image is that you hold out a pixel from the network, and you ask the network to predict the value of that pixel given the surrounding pixels. The reason this works is that if you imagine noise as independent, then the expected value of the center pixel conditioned on the surrounding pixels should be something like the denoised value. And what we did is we designed a graph-based version of this blind spot network, which operates on the voltage graph. So that gray graph that I showed you before, we designed a version of this network that operates on graphs rather than on pixels. And the advantages compared to like denoising the raw movie are pretty significant. So for one, the training and inference is much faster. So our networks take about 5 minutes to train on 10,000 frames versus about 24 hours if you're denoising the movie itself. And this whole technique, everything that I'm talking about today, is equally applicable to random access or projection microscopy methods where you might never actually create a voltage video. Instead, you might be taking linear measurements of your underlying signal. There are two simple operations that we implemented to get our blind spot network working. The main idea is that we generalized this pixel-based blind spot to a graph-based blind spot. And there's a spatial blind spot module which combines information across neighboring nodes of the graph. So you can imagine taking one node and trying to predict the value at that node from the connected nodes in the graph. And there's the same thing that happens in our temporal module. So we try to predict a held out time point given the values at adjacent time points. Now I'm going into the results, and I'll first validate just the network by itself. Here's an example of denoising at the soma. The blue little speckles are the observations. Red is ground truth, and green is the neural network output. Now here's the same thing at a dendrite, and the SNR is much worse at the dendrite because the spike has gotten truncated or filtered, and the noise level is the same. But you can see our network is still able to do a good job here. We also compared to a simple state space model baseline on single compartments. Okay, now I want to get to some simulated results. The prior slide was just validating the network by itself, and now we want to validate the whole pipeline. Can we go from a voltage movie all the way back to a 3D estimate, and will that work? And what we did is we generated multi-compartment simulations, and we used those to test the method. And on the top left, I've taken a bunch of nodes from the graph, from our anatomy graph, and I'm going to analyze the reconstruction on those nodes. Those nodes are blurry in the movie, and we're going to analyze whether we can still reconstruct the voltage. So this heat map here and those traces show the voltage starting at the soma and going away from the soma. And if you just naively use the fluorescence of the voltage movie, you make a big error where the nodes are farther away from the focal plane. So this little part here, this little panel, is showing that the dark blue nodes, or the bottom of this heat map, are farther out of focus. And that's where you tend to make a big error compared to the true value. When we run Dendro, I'm first showing just no neural network, just the output of the linear inverse problem. We get a noisy version of the truth, or it's at least close. And then we run that through our neural network, and that cleans up our estimates quite a bit. Now we ran a series of simulations with different focal planes, and we quantified how well the estimates match the ground truth voltage. If you just use the voltage movie fluorescence directly, that's here. And if you use Dendro, you do a much better job. The R-squareds are around 0.95 or so, even when you're far from the focal plane. So even when you're like 25 microns from the focal plane. Okay, I'm running real short on time. Here's a video. I think it's going to go. Okay, this is showing the two views of the true 3D signal and our estimated 3D signal. And I'm going to breeze through this because I have 1 minute. Now, I think a big thing that we're proud of is that we were able to run this on some real data sets from Adam Cohen's lab. So here's a result on one real data set. Again, I've taken the anatomy, and I'm tracing out a few nodes from the anatomy graph. And then this panel here is showing you how far those nodes are from the focal plane. The focal plane is the red line. And depending on the node, these are anywhere from 10 to 20 microns away from the focal plane. And now I'm showing our estimated voltage signals before and after we apply this neural network. And this cell is doing an interesting thing. The first two spikes here don't propagate all the way into the dendrite, or rather, they propagate passively. But it's on the third spike that the dendrite is engaging voltage-gated channels and performing an active dendritic spike. And this is something that our collaborators saw in their data, and we're able to now analyze it not just for the in-focus branches, but hopefully for the entire cell. I have another cell, but I'm going to breeze through it and show a video. The top is the original voltage movie. The second panel is PMD. That's this SVD-based denoising. Now I'm showing the estimated movie. That's what happens when you take our voltage estimate and pass it through the microscope model. So ideally, estimated is a denoised version of the original. Now I'm showing this voxel, this 3D voltage estimate. I'm showing two views, like a top-down view and a side view. And I'm also showing you the residual between our estimated frame and the original frame. And ideally, there would be no spatiotemporal structure here. There's a small amount. We think that's because of slight errors in our microscope model. OK, now our long-term vision for this is to automate fitting of biophysical models. The idea is that if you know the voltages across compartments, you can start to back out things like channel conductances and channel kinetics. In order to do that, we need to take our model that we have so far and add a model of the voltage indicator. Everything I've shown you has been in units of fluorescence. And in order to do this, we need to work with real physical units of voltage and then pass that through an indicator model to get back to the observed voltage movie. So I'm out of time, and I want to acknowledge our collaborators. So this has been a great collaboration with Adam Cohen's group. All the experiments you saw were done by Pojong Park. And I had computational help from Amol, Uku, and Chase. And I'm excited to hear, hopefully we have time for one or a few questions.
Are you able to detect some threshold events?
Yeah, we are. So I'm going to drink some water. These data don't have that much subthreshold because these are slice experiments. The cells are pretty quiet. We have other experiments where the optogenetic stimulus is less strong, and you can see clear subthreshold wiggles. Whether you would be able to detect, say, like a single synaptic event, I really doubt. But we can certainly see some subthreshold events. Yeah?
Would it be too ambitious to try to reconstruct large-scale recordings?
Can you use your seat mic, please?
Sorry. Would it be too ambitious to try to reconstruct from large-scale recordings using similar techniques?
Large scale meaning?
Neural pixels or a cross-shank?
I think Liam said Scott is working on some sort of biophysical modeling from neuropixels, but I don't know anything, so you should talk to him about it.
Okay.
Sorry, you should talk to Ian C., who's given a poster on versions of that.
Cool.
Tomorrow, probably. I wanted to ask you a question, though. It's kind of related. So the vision that you laid out was first denoise and smooth the voltage, and then fit the biophysical model to it. Could you imagine doing that end to end here? Why not just
Yes,
string
I
that?
really want to do this, and I've been bothering Liam that we should do it in an end-to-end way for about a year. It's very hard to do that. The reason we went with this way for now is that in these biophysical equations, there are many, many unknown parameters. And so we chose this method that's somewhat more, I want to say unbiased, although I'm using that word in a fuzzy way. We chose this model that's somewhat more non-parametric in the hopes that it'll get around the unknown parameters. Mm-hmm. We think that once we have this model, we can use it as an initialization and fit end-to-end using Jaxley, which is like a multi-compartment simulator. That's the dream, and we should chat more about that. But I agree, like the biophysics are there. Surely we should use them.